I have the incredible pleasure of working with our network of advocates across the country, of which we have about 150 on the go at the moment, talking to their local MPs and senators about funding for type 1 diabetes research. And we struggled for a while trying to get our messaging right for what we wanted all the advocates to say, going and talking to their MPs. And really, at the moment, in any given city, there is a young child practicing with their parent at the moment for meetings that are occurring this week and next week. And their basic message is, we want to find a cure for type 1 diabetes, we won't rest until we do, and research holds the key. So I think we are very lucky in that we have an organisation that has a really clear mission. We all want to find a cure for type 1 diabetes, and we all know that the way that we're going to get there is through research, and we're really very lucky that we have such engagement from the research community, from our broader community, from supporters like you, in showing that determination in delivering that upon that kind of research message. We're really hopeful that we can extend the fabulous work that is already being done and really build upon the wonderful foundations of the work being done by researchers like those that you'll hear from this evening. And we're focusing really on 3 different areas in our research asked to government, and they are on the areas of trials. We need more trials, talent, we need to have the best type 1 diabetes researchers attracted to our area of research, and translation, because at the end of the day, JDRF, as you know, is an organisation that is completely focused on patient impact and outcomes that actually are real and meaningful to our community. So happily, that's a very easy reason to remember: trials, translation and talent. And that's something that seems to be, we hope, resonating with government.
And thanks again to all of you for making time tonight to come and hear about how your contributions make an impact on the progress of type 1 diabetes research in Australia. Because it really is up to all of you and all of your contributions to drive the research progress here. Whether you're a regular donor, a major donor, a board member, a SLG member, an industry partner, we've got Abbott in the room as well. I just want to really acknowledge each and every single one of you in the room contribute to the research, and regardless of how much you donate or how often you donate, it's the collective, the sum of the parts that really makes the difference to the research that we've done here in Australia. Half of that is understanding where the gaps are and how we can respond to the community's needs across the whole life of type 1 diabetes, whether it's preventing type 1, and we've got a fantastic trial happening across Australia called the India trial, and this is looking at how environmental exposures happen during pregnancy and in the early life of a baby, and how that can potentially impact the development of the autoimmune disease that then leads to type 1 diabetes. And so this is really about how can we prevent type 1 in future generations. We also, and that's also to mention, I guess, and acknowledge the work of the Walter and Eliza Hall Institute here in Melbourne as part of that trial. We also work throughout the lifespan of people living with type 1, whether it's related to glucose control, and we'll hear a little bit from Christoph about how we can improve insulins, and particularly around smart insulin, how we can also better educate and produce guidelines for healthcare providers and also families and people living with type 1 in relation to the impact of diet and exercise on glucose control, as well as technology. So we've got fantastic closed loop trials. One of the other things that JDRF does that's really exciting is looking at how can we address some of the key challenges that face research progress today. And one of those challenges is the next generation of researchers are having to figure out how to be better communicators, better grant writers, how to talk to government, how to engage in the industry. These are all very important aspects of research that isn't necessarily taught at university. So we've developed the Future Research Leaders program here in Australia, It's a world first in terms of what we're incorporating into a research leadership program. So we're bringing together experts in leadership training. We're linking the next generation of researchers to senior current research leaders in Australia and globally, as well as providing them access to some of our networks to get them better educated and more experienced at talking to some of the key stakeholders, including patient communities.
I think one of the greatest things that we do is we are building talent in the area of diabetes. We have to compete against a lot of other areas in medical research, and historically Australia tend to be more interested in cancer and some of the other diseases. But fortunately, with the support of JDRF, we continue to produce great new scientists in the field of type 1 diabetes. And what we believe is that the problems in diabetes, particularly glucose, and elevated blood pressure, which often occurs, and the gaining weight, which we often see with people who are on insulin, leads to all this damage. And there are targets that we have to block or treat so we can reduce the major complications of diabetes. In particular, blindness, amputation, kidney disease, which can ultimately, unfortunately, lead to dialysis, and cardiovascular disease, which is relatively underappreciated, but unfortunately, cardiovascular disease remains the major cause of death in type 1 diabetes. And historically, people with type 1 diabetes have reduced lifespan, but fortunately, one of the great areas of progress in the last 20 to 30 years during my career has been the dramatic reduction in these complications. Although they're not gone, we've reduced the burden at least 50%, which is much more than many other diseases that are widely around in the community. And so it's very important to reduce the heart attacks, strokes, these others, which are very important for the quality of life and lifespan in diabetes. So for example, I'm just going to give you examples of targets. So one of the targets is we get a lot of what we call oxidative stress in diabetes. For some reason, high glucose causes oxidative stress, and we know there are a whole range of causes of what we call oxidative stress or reactive oxygen species. And in particular, we've identified an enzyme called NADPH oxidase, which we've been able to show initially in animal models, if we block it, we can reduce complications. And with the support of JDRF, we have commenced a clinical trial, which is occurring in the next building on the 4th floor, where we've recruited, we're going to recruit 140 patients with type 1 diabetes and early kidney disease, and some of them will receive a particular drug which blocks this enzyme. And this work is being supported by JDRF. These trials are expensive, $4 or $5 million, but we've been very fortunate as part of the initiative with the federal government and JDRF, and already the first 10 patients have been recruited in 2018. We also know there are other reasons for the complications. So this is a protein which is being bombarded with high glucose because of diabetes. And that glucose then turns into these red spots called AGEs. They bind to the protein. That protein then binds to what's called a receptor sitting on the cell, and then that causes all these other proteins to be produced, which cause complications. So one of the areas of interest that we're very interested in is inhibiting this protein called RAGE. And we've made a very exciting discovery, just patented 4 weeks ago, where we have a new way to block that interaction. And hopefully, if we can prove in the animal models, we already have early results that are very positive, we could produce a new drug which could actually inhibit these complications. And it's very important because these complications, the eye, the kidney, the blood vessels, they're all very similar. So if you can find this unique inhibitor, you'll be able not just to treat one complication, but hopefully 3 or 4. So we're trying to work out how to stop that scarring. Because if you stop that scarring, the kidney will not fail, and then you won't need dialysis. And here we've produced a peptide. It's a 12 amino acid protein, and if you give that to the animals, you dramatically reduce the scarring to almost the same level as in a non-diabetic. And we're now trying to optimize that molecule so we can actually produce something that we could give people to stop their kidneys from scarring. For example, Everyone talks about genetics. How does genetics influence diabetes or its complications? And what we now know is it's not just the genetics, it's epigenetics. Epigenetics is the link between the environment and your genes. And what we've discovered is that glucose or sugar is a very powerful modifier of these genes. And this is the study of epigenetics, and we've been studying this in diabetes. What we do is to emphasise that we're doing a lot of exciting work trying to find a cure for these complications. We've got very good results in some of the animal work. We've already started the clinical trials, and we have some new targets that will also be very interesting as well.
I am one of the professors there in his newly established department, which is dedicated to understanding and preventing the complications of diabetes. So of course I work on the eye, although originally a kidney researcher, and I just want to highlight to you again, you probably realise this, the growing burden of diabetic retinopathy and its incidence, as shown here very nicely on the graph here. You can see that we already have a great many people who have the early stages of diabetic retinopathy, but also in the small circles on this graph you see this increasing number who have the vision-threatening forms of the disease. So it's this very progression of disease over time. So it's expected by the year 2040 there could be as many as 70 million people. So that's a statistic we don't really want to see. And of course I don't need to tell you it is a leading cause of blindness in people of working age when people are trying to be the most productive. Little blood vessels in your retina, which is at the back of your eye, and it's very thin, only a couple of millimetres thick, and really like a piece of tissue paper, if you can think of it like that, but highly complex, has these little blood vessels which become damaged due to high glucose, high blood pressure, and high lipids and many other things that Mark's talked to you about. But unfortunately, what we've now learnt is that it's not perfect, and 50 to 65% of eyes don't respond. So we come back to this issue of prevention and understanding more about actually what's going on in this very complex but very beautiful little tissue that we really need for our lives. We developed, actually it's a spin-off from what Mark did, looking at ACE inhibition and lowering blood pressure, and we worked on that, and we were the first group to show that lowering your blood pressure can help stop the blood vessel damage in the eye.
As Julia mentioned, we have a glucose sensor which senses glucose continuously, which is talking then to an insulin pump which delivers insulin depending on the demand. So it's like sitting with this guy sleeping in his Tesla and driving at 130 on the road. I wouldn't recommend that. But there's still one inherent problem, and that's because with the injection or with the pump, insulin is delivered subcutaneously. And then it takes time for this insulin from the subcutaneous phase to go into the blood. So there's always a delay between the injection of insulin and when it actually hits the target. And that's probably best shown here in this really nice review from a couple of years ago. When you look at what's the actual physiological demand of insulin after a meal, with 3 meals a day, and what current insulin therapy can actually provide. You see, it's never fully in sync. It's always a bit behind, or a bit too low, or a bit too high. And that's caused, of course, issues with hypoglycemia. So we turned to nanotechnology to maybe have a particle which can detect glucose and is loaded with insulin, and then in a glucose-dependent manner can release insulin and also stop releasing insulin. So what's nanotechnology? So nanoparticles, by definition, are particles between 10 and 1,000 nanometer. So if you look here from the scale, they're pretty much a little bit bigger than a glucose molecule, but they're smaller than a virus. So that's where nanotechnology fits in. They can be composed of natural or synthetic polymers, and because they're similar to the size of a cell or something like that, or a virus, they can actually mimic this function quite nicely. So the two, on the nanoparticle GRI space, there are 2 principles how you can achieve this. So one is, and there's a lot of preclinical publications already out there, of particles which have insulin inside, and they can swell and they can shrink depending on the glucose environment. So if they swell, the insulin will diffuse out. If they shrink, the insulin release will stop. We don't think that it's a very good approach, because it's a lifelong treatment. So with this approach, you basically end up with empty particles which are in the body forever, and this can cause potentially side effects over multiple years. So you need something which basically, after the insulin is delivered, completely disappears. So you need a particle which erodes from the outside. So you have insulin packaged, and then the glucose comes in, the insulin is released from the outside, and then once the particle is completely used up, it's gone. So how can we make these particles? It's just one example how these particles can be made. It's called the layer-by-layer approach, where you have insulin somehow packaged in the middle, and then you have different forms of glucose-responsive polymer on the outside. And you can fabricate that manually or automatically. And here on the right, you see how these particles look like in electron microscopy or in fluorescence, different sizes. So we can make them very, very homogeneous and with very low polydispersity. We use a polyphenol, which is this group here, which is actually quite a very health-promoting chemical entity. It's an antioxidant. And this polyphenol we combine with also a non-toxic group called borate. And these borate and this polyphenol, they form a bond which is inherently glucose responsive. So when you form this bond and glucose comes in, this bond will break. And that's the principle of a glucose responsive insulin system in our case. And just some data which we published a while ago, we can make these particles, and then when we come in with the sugar, you see that we can degrade these particles and dissolve them with a model insulin construct. So that's basically where we are at, and we would like to have an oral formulation ideally. So the dream and the holy grail is really to have a tablet you take once a day, and then all your insulin requirements are met, and they're automatically like an autopilot dealt with.
So what I'm going to talk about today is a very surprising discovery which we made in my group a couple of years ago, that will allow us to target type 1 diabetes right at its core. So it's the goal to regenerate beta cells, and I want to be upfront here. We would have never had the opportunity to follow up on that surprising discovery if it would not have been for JDRF funding that we were able to attract and also move with us to Austria. So one thing that I have always been really fascinated since I was a child is really that our body consists of an unbelievable number of cells, right? About 10 to 20. We actually don't know how many cells there are in a body. It's a lot. And as you have seen also from the other speakers, cells are very diverse, and they have different functions in different organs, but there's really sort of one system of checks and balances which really applies throughout the body. And this checks and balances system regulates the amount of growth and the amount of death in cells. So as you know, a dysregulation in either direction of this balance is equally fatal. So too much growth is typically linked to disorders like cancer, so we don't study those. This is potentially also relevant in the context of what we do. And then too much death is linked to degenerative disorders, and we focus our efforts entirely on type 1 diabetes. All right, so what is important in the context of this balance is that it is typically one particular group of cells that are affected by this excessive death. And as you know, in type 1 diabetes, it's beta cells in the pancreas that, among other things, are responsible for regulating insulin levels. So this property that it's one group of cells really sort of challenges how we approach the problem of regenerating or restoring cell death. Because I'll just go one slide back, because the last thing we want to do is push other cells in the body into too much growth in our attempts to regenerate cells that suffer from too much death. So we have come up with a hypothesis how we could do that. And this idea would be, and it has been actually around in the diabetes field, sort of as a theory for a while, is if we were to have a tool which would allow us to locally activates survival and growth of beta cells, and maybe also in a temporary, so in a time-controlled manner, then we could really tackle the disease right at its core. This is a quote from a book from a colleague from Mount Sinai, and this has sort of been in an idea and the hope that we have found a way to actually put this into research. All right, so what did we discover? So we discovered in my previous work that beta cells, very surprisingly, and I'll be happy to explain why that is and what the genes are that are involved in more detail afterwards if you would like. We found that they're actually light sensitive. So the sort of coupling light to survival is something that we are very familiar with maybe from the plant world, but we found that beta cells do that as well. So you can shine light on them and they start to grow more robustly. I'll just show you on the last slide a bit of data that substantiates this. So we looked at this both in tissue from mouse and as well as in tissue from patients from donor tissue. In all of these cases, you see that the light-treated cells, which are the green markers in this particular diagram, grow much more robustly than the cells that haven't been light. So we're currently translating that in a variety of ways, including animal models and more data on human cells.
Oh, Harold, well, firstly, thank you for a remarkable presentation. Some of us have been around for a long time, and it's nice to see things really starting to happen. In the light regeneration, I assume you have to sort of get beta cells out to stimulate them, and then you inject them back.
Thank you very much. That means a lot. So we envision two potential ways to translation. One is we can make beta cells that are outside of the body proliferate. We talked about transplant strategies, so we could potentially multiply cells to have more cells for transplantation or make them more robust. And then the second option that is independent of that would actually be to do it in the body on site. And this is what we are currently looking at using mouse models.
So I assume then, if you take the beta cells out, get them to regenerate, you can do that on a continuous basis, and then putting back into the body, there'd be no rejection. So,
yeah, that would be, I think, that would be the vision, sort of, right? That you sort of be exploring how hard can we push this? Is sort of infinite regeneration really? So, and we're fairly confident that some of our colleagues in our field will take care of some of the things that relate to what we do, so we can focus on things that I've shown you. Because it's really, like you said, I think this lives of collaboration.
I think the problem we've got is that JDRF was on an upward climb for raising money, which was very good. We didn't have that many researchers working in the area, and we recruited lots of great people like Harold who come from other areas. And what's happened is we've stalled, and the problem is it's we're hit, because what's happened is the funding is either stabilised or not continuing to go up, but the research is getting more expensive. So what we used to do is like simple things, like give a drug to a few rats or mice, now is sequencing the whole mouse, then getting some clever nanoparticle, needing chemists over at Melbourne Uni to send us agents. It's getting very complicated. The work is getting very complicated. The funding rate is dropping, so that what used to be a 25% funding rate is now down below 10%. That means for every 10 grants you write, you're lucky if you get one. So you think if a grant takes a few weeks to write, you've got to write 10. Half your year is writing grants. When are you getting the time to cure diabetes?
I think compliance is also a big issue. So JDRF was organizing a forum last year in New York around glucose-responsive insulin, and there were company representatives as well. And they were actually talking to the FDA. And the notion from the FDA was that we have diabetes under control in a way. We have short-acting, long-acting, we have pumps and things like that. So if at the top people don't see that they still need to develop things and to progress and cure, then it's really a problem. I think everyone on every level of government has to understand that it's not there that we have everything under control.
I agree with everything that's been said, but one of the things that does concern me are the young people. You saw we've got some young, bright people in Australia who want to work on type 1 diabetes. But in the current funding climate that we have in this country, they may not stay with us and they will go to other areas, completely different fields. And you hear about this, but it's a reality. And so we are pressed to keep them. Now, it's wonderful to hear that JDRF Australia have got some mentorship programs, and this is fantastic. But the problem is they have to eat. We all have to eat, actually. They have to eat. And that really frightens me. I've never seen it before like this, and I don't want it to continue. So I think we need to think a bit more about having more fellowships, honestly.
Harold, can I just ask you, in terms of your experience overseas, what can we learn from other countries, actually in Europe, regarding supporting type 1 diabetes research?
Yeah, this is a hard question to answer because I think Australia is doing a pretty good job, to be honest. I mean, I think Australia is one of the few countries who has a JDRF activity of their own. I think it's very few, maybe 5-ish, 10-ish in total. So as far as I can say, and this may make sense why we're here, right? That actually we think things are great here, and it is collaborative activities. It's if you think about the research landscape in a city like Melbourne, there's very few areas in the world where you have this density of scientific excellence. Maybe Boston, Greater Boston area, MIT Harvard, maybe San Francisco Bay Area, Berkeley, UCSF, Stanford. But I would be hesitant to say that Germany or Switzerland are doing things much better than type of research. You might have to say.
Jennifer, you talked about the treats that you're working with, and you talked about their improving the damage to the tissue in the eye. Are they actually, are you seeing that the tissue is repairing or it's regrowing, or is it just preventing the damage?
It's preventing. It's a good question. So what happens? The idea is that these cells, these Tregs, are made in the body, and we want to give agents, whether they be even pharmaceuticals, but also dietary interventions actually can work. We're working on some natural things that you give early that make them, it takes a little while for them to build up to numbers, and they migrate into the eye and they prevent the damage. It's like with all the complications. Once you have very severe damage, you're in it's a tough place to treat someone who's got very severe eye disease. You can be done, but it's better always to prevent. That's not to say it won't work also when there's already established damage. But the idea I really think is the best way to go is if you have, say, a young person who's just been diagnosed with diabetes, for example, to give it early, something that's gentle, that's not has side effects. And so one of the things that JDRF has funded me for is to look at a particular nutrient that triggers the Treg as a component in the nutrient that actually makes those Tregs proliferate. So that would be really something.
I might just add to that, actually. That was a fantastic question. We recently awarded a grant up in New South Wales to Shane Gray, who does a lot of collaboration with researchers down here in Melbourne as well. He is actually working with a junior researcher at the Victor Chang Cardiac Research Institute. And what they've found actually is very similar to what Jennifer is working on, is that this particular immune cell could actually be assisting in repair. So this researcher has found that these little Treg cells in zebrafish, he's looking at, I believe it's spinal cord regeneration. And so he's found that these, in zebrafish, if you damage the spinal cord, these little Tregs come in and can actually repair the damage to spinal cord. Shane Gray fortuitously went to a talk and heard him talk about it and said, why can't we look at whether Tregs can actually go into the pancreas and repair beta cells? So we've just invested in innovative research grant up there to look at these exact same cells, and it's a very similar process. So hopefully, potentially, it could be involved in the care as well as preventing the damage in the first place.
Well, I just wanted to relate an experience that my daughter has had serious eye problems, and the latest version of the injectables has actually improved her eyesight, particularly one eye. So it's just not preventing. So it's quite a remarkable achievement. And I'm also particularly pleased to hear that red wine's good for my eyes. My experience is quite the reverse.
Moderation in all things. I think that those drugs, of course, do work. It might work in one eye, not particularly well in the other, but they're amazing.
That's exactly what's happening.
Exactly. But it's the other 50% for people that don't, where it doesn't work. And they work also very well, they work better actually in age-related macular degeneration. You know what that is when you have vision loss as you get older, something to look forward to. When you get older, you have a sort of a vessel damage in the back of right at the back of your eye. That's very pleasing to them if it's worked at this time.
If it wasn't for JDRF, where would you be? Where would your research be?
I'd be at home. Seriously, I think one of the big advantages has been historically for one of the groups is that JDRF provide a lot of critical early funding. There's different criteria to, let's say, NHMRC. They're more interested in application and impact. They rely on less preliminary data, but a more brilliant idea. I think Harold's idea is really brilliant, and this is getting innovation, JDRF funding, the immune cells in the eye, all these projects, I'm not going to my projects, but the other four are all highly innovative. They will end up getting government NHMRC funding, but they may need that early JDRF funding at the start. The other great thing, of course, is the JDRF does clinical applications. So if Jennifer's got this great discovery, or Sharon has about these gels, you will be able, NHMRC will not have the money or the interest in doing the big clinical trial. They will not find $4 million to do the NOx inhibitor in the type 1 diabetic patients. So I think that's what JDRF does. The other thing is JDRF gives researchers that work in type 1 diabetes an edge, because we're competing against cancer, neuroscience, and whatever. Without JDRF, type 1 diabetes would be on the bottom of the list for NHMRC, because it's not as big a clinical problem as dementia or Alzheimer's or cancer. So the government is always interested in major impact. But type 1 diabetes is one of the commonest conditions in children, and the public needs to support it. JDRF gives us the chance to research into that area.
And so our project we started this year, but already, so I suppose it's enabled us to develop a research team. It was a concept call, so it is like an innovative brand, big picture, new therapy. But already we have 3 students on the project. So one PhD student who we recruited from Western Australia, a master's student and another undergraduate student. I think picking up on Jenny's point, this will allow us to actually mentor these students and then hopefully they'll be the next future leaders to carry on with research. So it does make, in even a short period of time, quite an impact.
Yeah, so for me and for Frank as well, we would never be in diabetes research without JDRF, because we're not in this area at all.
Wow. Why am I here? I'm here because these guys inspire me, and the search for a cure is something that we all relate to so strongly, isn't it? It's something we want for our children, it's something we want for our friends with diabetes, and it's something that many of you in the room are journeying through every single day of your lives. And I remember Paul Zimmet saying to me one day, you never get a day off from diabetes. And whenever I repeat that to a parent with a newly diagnosed child, they said, yep, you can't be tired, you can't be disinterested, you can't be busy, you never get a day off from type 1 diabetes. And having a child with type 1 is a hard road. So it's fabulous to see all of you here tonight to hear the wisdom and the learnings and the practical application that is happening here at Monash University in conjunction with the translational team, I guess too, at Baker IDI. And we have always, as a country, punched above our weight in medical research, and tonight you've got some very clear evidence as to why that is still so today. So thank you for coming. Thank you to those of you who engage with JDRF. Some of our leadership team are here tonight. Our staff team are also here. You can help in all sorts of ways. You can help by being an advocate, as Saz has suggested to you tonight. You can help by talking to your friends and family about supporting our events, coming to our galas, doing our walks, doing the crazy, wonderful things that we try each year to raise extra money for research, because we know that there are people who can make a difference. It's really important, and we thank you for what you're doing.